is a PowerPoint presentation about the lymphatic system. At this time of your study of medicine in the first year, it's important to keep calm and study in a smart way to become a doctor. And we should remember that at this point of time, we are talking about the basics. Here is a diagram of the circulation. The heart in the middle, and out of the left ventricle there is the systemic circulation, and out of the right ventricle is the pulmonary circulation. At this area we have what is called the microcirculation, the capillaries between cells. Capillaries start as an arterial capillaries, and then as the exudate interacts with the cells will take whatever they need and they secrete whatever they don't. And then the interstitial fluid is going to be drained by the venous capillaries. This process is very, very efficient in normal conditions, but we have a problem. The problem is that we have large molecules produced by the cells, or we have foreign bodies like bacteria. These structures, they cannot get into the venous side of the capillaries. Therefore, we need a system that will pick up these large molecules. And this system is the lymphatic system. The lymphatic system is starting with blind-ended capillaries. These are not capillaries like the arterial or the venous. They are special capillaries. They can pick these large molecules and then they drain them. When they drain them by blood, by lymphatic vessels, they need to be filtered by lymph nodes. And after they are filtered, they return back to the venous circulation. Therefore, the lymphatic system is there for large molecules, for bacteria and other large structures. Lymph nodes are the structures that will receive the lymph from the tissue, and they are filters. Then the whole system is going to end in the venous system. So it starts as blind-ended vessels between cells. They are picked up by vessels. These vessels empty into lymph nodes. Lymph nodes will filter the contents, and then the whole thing is going to end into the venous system. This is another example of the microcirculation, and it is important to ask so many questions. Here is the arterial side of the capillaries between cells, and after the interaction, the venous side of capillaries will take the outcome, and the venous capillaries will join the venue. Again, I have to stress that this is a very efficient procedure. it takes care of the tissues. Here is a three-dimensional diagram of the area of microcirculation, where we can see that the lymphatic vessels start as blind-ended structures between the cells and the capillaries, and then they join together to form large lymphatic vessels and join the lymphatic system. This is a diagram showing how special the lymphatic capillaries are different. They have the ability to allow large molecules to pass between overlapping endothelial cells. These endothelial cells, they are not joined by tight junctions or any other type of intercellular junctions you have studied when you studied the epithelial tissues. These endothelial cells, they overlap, and a large molecule can get into the lymphatic vessels by passing between the overlapping endothelial cells. This is a very closer look at the beginning of the blind-ended lymphatics, where we can see here inside this black oval shape the overlapping junctions between endothelial cells, and the large molecules can get in easily while they cannot enter a vein. And to keep these lymphatics opened, we have what is called the anchoring filament, kind of connective tissue that attaches to the endothelial cell and to the surrounding tissues, so that when pressure inside these tissues increases, these capillaries will not collapse. They will stay opened because of these anchoring filaments. In the first diagram, we can see that the arrows are indicating that there is increased pressure around this blind-ended lymphatic capillary. And in the second diagram, we can see that large molecules and excessive interstitial fluid can pass in because of the overlapping junctions. And then in the third diagram, we can see that this group of large molecules and excessive interstitial fluid will pass, and there are valves. Lymphatics have lots of valves, so that there will be no backflow of the lymph into the tissue again. Here is another diagram of the blind-ended lymphatic capillary. And then as they collect large molecules and excessive interstitial fluid, there will be a flow of the lymph inside these vessels, and there are valves. There are many valves, so that there will be no backflow of the lymph to the tissue again. This is a histological section of a lymphatic vessel, where we can see that there are valves, and we can see the contents of the lymphatic vessels which is excessive interstitial fluid that the veins could not drain. These valves are made simply of 2 layers of endothelial cells, and they are very efficient, and they prevent backflow of the lymph into the tissue. One more diagram showing the blind-ended lymphatic vessels, and these capillaries, they join and form larger ducts, lymphatic ducts, and then they form even larger lymphatic vessels, and they are full of valves. In this diagram, in which we can see that the blind-ended lymphatics will pick up excessive interstitial fluid and large molecules, and there are many valves, and these lymphatics end up in a lymph node. A lymph node is a filter for the lymph, and the lymph can pass through one lymph node, and in other instances it passes into 2 or more lymph nodes, and the story ends that the lymphatic vessels will empty into the venous system. After collecting the lymph from the interstitial fluid and passing through the lymphatic vessels, carrying antigens, large molecules, they enter the lymph node. The lymph nodes, they contain lymphoid follicle, and these can also be called lymphatic follicles. What is a lymphatic follicle? It is a group of lymphoblasts. Underline the last part of lymphoblast, meaning underline the blast. These are dividing cells. They mature into lymphocytes, and they come from the bone marrow, and can see that they are pale because the lymphoblasts are large cells. They contain more cytoplasm. This is the basic unit of the lymphatic system. Now, if you look at the periphery, you will find that it is darker in color. These structures that are dark in color are lymphocytes. The mature lymphocytes. These lymphocytes, they have large dark stained nucleus with little cytoplasm, therefore they appear as dark. This lymphoid follicle is a primary lymphoid follicle because it has not been stimulated by an antigen or bacteria or any foreign material. These lymphoid follicles are present in so many tissues. They are there to produce lymphocytes, and lymphocytes will produce antibodies to attack antigen. Now, these lymphoblasts, I will repeat again, they come from the bone marrow and they live inside tissues, and they are ready to produce lymphocytes which will produce antibodies. Here is a lymphoid follicle. The center is pale and it is called the germinal center. What is it made of? It's made of lymphoblasts. Now, this lymphoid follicle has been stimulated by an antigen. Therefore, we are going to see that these lymphoblasts have differentiated into lymphocytes at the periphery. It is rather thicker area than the image before, and these lymphocytes now they make a thicker layer around the germinal center, and it is called the marginal zone. Here is a cross-section in a lymph node where you can see several lymphoid follicles. This one has a germinal center, and you can see that it is surrounded by a marginal zone, which are mature lymphocytes producing antibodies. And this is the marginal zone. In this example, the germinal center is smaller and the marginal zone is much thicker. That means this lymphoid follicle has been stimulated to produce lymphocytes, to produce antibodies. Now, when this lymphoid follicle is stimulated, it is changing its name from primary lymphoid follicle into a secondary lymphoid follicle. Here is a more example of a lymphoid follicle where you can see that the pale area in the center is the germinal center made of what? Made of lymphoblasts. And these lymphoblasts, when stimulated, they produce lymphocytes, and this is the marginal zone surrounding the germinal center, and it is darker in color because of collection of mature lymphocytes. Here is a famous example of cross-section of the vermiform appendix, which is famous for having lots of lymphoid follicles. And I'm sure now that you know where are the lymphoid follicles. Here is another example of a lymphoid follicle with a germinal center and a marginal zone. This is a diagram of the small intestine where you can see that in the submucosa as well as in the lamina propria there are many lymphoid follicles. The ones that are filled with the blue dots are secondary, they are stimulated, and the ones that have empty center are the primary ones. It is important to write down every now and then notes to check our understanding. Lymphatic vessels are blind-ended tubes. They have thin endothelial walls. These lymphatic capillaries, they are not like blood capillaries. They are only made of endothelial cells, no basement membrane. And it is only a single cell in thickness, and They are arranged in overlapping pattern. There are no intercellular junctions, and they join to form larger network of tubes, and these tubes or lymphatic vessels will join each other, forming progressively larger lymphatic vessels, and at the end of the day, near the large veins, they form two lymphatic ducts, which we will see in a moment. Here is a diagram of the filter of the lymph node. The lymph node is going to receive multiple afferent lymphatic vessels coming from tissues, and the lymph will enter the lymph node in a sinus surrounding the lymph node, and this is called subcapsular sinus. In lymph nodes, lymphoid follicles are having a capsule. This is a group of lymphoid tissue that is capsulated, while lymphoid follicles, which are widespread into tissues, they are not encapsulated. Now, after subcapsular sinus, we reach the outer cortex of the lymph node, which contains lymphoid follicles. As you can see and go around, you can see that some of these lymphoid follicles are primary and some are secondary, and this outer cortex is predominantly filled with B lymphocytes, lymphocytes that produce antibody. Then we go to the inner cortex, the lighter green area, which is predominantly filled with T lymphocytes. T lymphocytes are different from B lymphocytes. B lymphocytes produce antibody. T lymphocytes are killer cells. Then we have medulla. It's the place of collection of the B lymphocytes, the T lymphocytes, the filtered interstitial fluid, and the result of interaction between the B and the T lymphocytes and any bacteria or foreign body. If the B lymphocytes and the T lymphocytes have dealt with the bacteria, then there is an interaction, there's a battle between the two, and the lymph nodes will become inflamed and become larger. If the bacteria won the battle, then they will come out of this lymph node through an efferent lymph vessel. It is usually one, while the afferent are multiple. Lymph nodes, they are bean-shaped structures widely distributed throughout the lymphatic system, and they are providing filtration for the lymph, and the lymph is filtered before it joins the bloodstream. And it has two distinct regions, as we have just seen. It has a cortex and a medulla. The cortex contains follicles, their collection of B lymphocytes. At the center of the follicles is an area called the germinal center. These are predominantly B lymphocytes, and the remaining of the inner cortex T lymphocytes, which are killer cells. And there is one afferent lymphatic vessel. Here is a cross-section of a lymph node, where you can see that in the outer cortex it is full of lymphoid follicles in the form of circles, and the medulla has no lymphoid follicles and collection of connective tissue and lymphocytes leaving this lymph node. One of the lymphoid tissue that you can see is the Palatine Tonsil. These are collections of lymphoid follicles on both sides of the isthmus of the mouth, point of entry of food from the mouth into the pharynx. If there is any kind of bacteria or viruses, these follicles will get larger, and you see that the tonsils are large. If they are not exposed to any stimulus, they get smaller. This is collection of lymphoid follicles at the back of the nose in the nasopharynx, and this is for the pharyngeal tonsil. When it gets larger because of being stimulated, it blocks the nose. Therefore, the patient will become a mouth breather. Here is a section of the tonsils. You may say, where are the follicles? Here is one example of a lymphoid follicle where you cannot see a germinal center, no pale area in the center, because this has been stimulated and lots of lymphoblasts have changed into lymphocytes to produce antibodies. In this diagram, it is showing a special lymph node which is called the jugulo-digastric lymph node. This is the first lymph node to face lymphatic vessels coming from the palatine tonsils. Therefore, if the tonsils are inflamed, we should always palpate this lymph node, the jugulo-digastric. Now, where is this lymph node? As you can see from this figure, it is along the anterior border of the sternomastoid muscle near the angle of the mandible. Here is a patient with enlarged jugulo-digastric lymph node lying anterior to the sternomastoid at the angle of the mandible. And when this lymph node is losing the battle against bacteria, then the afferent lymphatic will take the bacteria to the next group of lymph nodes, which are the superficial and then the deep cervical lymph nodes. Lymph nodes are present all over the body. For example, there are lymph nodes draining the head and neck, and we have went through these groups of lymph nodes when we talked about the face. And then lymph from the upper limb, they drain to lymph nodes in the axilla. Lymph from the lower limbs, they are drained to lymph nodes along the inguinal area. And there are many lymph nodes in the thorax and in the abdomen. Each region has its own group of lymph nodes. What do we see in this figure? First of all lymph below the diaphragm. That means lymph from the lower limbs, from the pelvis, from the abdominal cavity, will collect into an area, dilated area of lymphatic vessel called cisterna chyli. And from this cisterna chyli, a duct that goes up in the thorax and then turns to the left and joins lymph coming from the head and neck and the left upper limb, and the whole lymph is drained into the left subclavian vein. Here it joins the venous circulation. That's lymph coming from below the diaphragm. On the right side, we also have lymph trunks coming from the head and neck and the right upper limb, and these trunks, they also end up in the right subclavian vein. This is a diagram showing how extensive the lymphatic vessels and the lymph nodes are in the body. These are the lymph nodes of the head and neck, and we have went through these groups, and they are preauricular, also called the parotid, the submental, submandibular, the posterior auricular, the occipital, superficial, and the deep cervical lymph nodes. These are the lymph nodes of the axilla, draining the upper limb and the part of the thoracic wall, and they are groups into lateral group, anterior group, central group, apical group, posterior group, and they all drain into the infraclavicular group of lymph nodes. These axillary lymph nodes are very important, especially in case of cancer of the breast, because cancer of the breast is a mass of cancer cells. Cancer cells don't produce good intercellular junction. Therefore, cells separate from the mass. They are picked up by lymphatics. So when we examine a lady with the suspected cancer cells, we have to examine the axillary lymph nodes, and we have to know the groups of the axillary lymph nodes. There is a problem, and that is cancer of the breast can spread to a group of lymph nodes posterior to the sternum, which we cannot palpate. So we cannot tell the whole story of whether this cancer is